Hello, people. I'm Jorge Bogantes, Natural Resources Specialist at the Anacostia Watershed Society. And here with me is John Pfeiffer, Curator of Bivalves. We're here at the National Museum of Natural History in DC. We are in the invertebrate collection. So John is going to tell us a little bit more about the collection, and then we're going to look at some of the cool mussel specimens they've got here.
Thanks, Jorge. Appreciate you. And thanks for joining, everyone. It's great to have you with us. So yeah, we're in the West Wing of the Natural History Building at the Smithsonian's National Museum of Natural History. And this collections area houses all of our mollusk specimens. So bivalves, your favorite group, but also snails and cephalopods. And on the other side of the collection over there are the corals. So this facility is just is storing the dry specimens in our collections. The specimens that are preserved in ethanol with like the animals inside the shells are stored in a facility in Maryland. So this is our dry collection, and there are millions of bivalves here. We have 250,000 lots, and let's go take a look over here, and I'll show you, kind of introduce how our collection is organized and what a lot is. So this is, the collection is organized in these cases, and they're movable, which allows us to kind of like an accordion to make them to fit more specimens. And then with each case, organized in these drawers, and the drawers are organized taxonomically by species. And so here we're just looking at some really beautiful freshwater mussels. These are scallops. So I thought I'd just introduce the collection by showing these really pretty ones.
Look at those colors.
And so, yeah, I said that the collection is organized into lots. And so each one of these boxes here represents a lot. And with a lot is just a particular species collected at a particular place at a particular time. So all these animals just kind of give you a representation of the variation found at a given place in a given time. And this type of organization allows us to understand how biodiversity has changed across time and space. This is like the physical, we are chronicling changes in biodiversity by organizing and collecting specimens like this.
And these are from the Philippines.
Yeah, and so within a species, they're organized geographically. So this is one species, and these are all from the Philippines. And these are some of my favorite drawers in the whole bivalve collection, because they're just remarkable color variation within a species, within a population, like at a given sampling locality.
And again, these are all scallop species.
Yeah, so similar to the scallops that you'd eat at a restaurant or in your home. And these species in particular, like they have a really large adductor muscle, which helps them swim. And so our dinner is relying on the evolutionary adaptations of these bivalves to swim. Like most bivalves can't swim, right? But because they have these really large adductor muscles, they have evolved the ability.
And that's what you eat when you eat a scallop, the adductor muscle, which is interesting. Many people don't know that.
Yeah. So unlike oysters and mussels, marine mussels, you eat the whole animal.
Yeah.
But for scallops, you're just eating that one really large mussel. But this right here is like my favorite drawer. One of my favorite drawers. I hate to play favorites, especially for marine animals, because the majority of my research is
amazing
focused
colors.
on freshwater. But yeah, these are just really remarkable specimens. They look like
And these are
crayons.
From where?
I think these are from Japan.
Japan. Oh.
I can't remember right now. But yeah, so that's kind of how our collection is organized. And show off some marine.
Yeah, now
bivalves.
let's look at some freshwater mussels. Maybe should we start with the oysters?
Yeah.
Yep. So we're gonna see some freshwater oysters. So the mussels belong to the Unionidae group, and then that group has 6 families of freshwater mussels. So we're gonna look at some of these families. Now here in the Anacostia River, they all belong to one family. But right now here, John is gonna show us some of the exotic families of the mussel, of the freshwater mussel world.
Yeah, so the Unionidae, like Jorge was saying, is found in North America, but it's also found in Africa and Eurasia. But these families that I'm about to show you only occur on the southern continents. And one of my favorite families, it's not a very species-rich family. I've only thought to be about 4 species, but are these freshwater oysters. So just like the oysters, if you're in this region from the Chesapeake Bay, these species have independently converged on a similar morphology where they physically cement themselves to the other side of their shell. And so they look remarkably similar to like an oyster that you would eat. And so these freshwater mussels are cemented to the rocks in like fast flowing rivers in the rapids, and where they spend their entire lives until someone plucks them off and puts them in a collection. Well, this is the family Etheriidae, and these ones are all from Africa here, or Africa or Madagascar. And you can see that there's just a lot of variation in the size. And if you've ever looked at like a unionid, the inside of a freshwater mussel in like the Anacostia, you might notice that the muscles that close the valves are much smaller, like they're just these little circles. But in this species, they're really elongate and much larger. Another adaptation to help them open and close their valves when they're attached to the rocks.
We could have perfectly had some of these in the Potomac Gorge, but wrong region of the world.
I would absolutely adore that. Yeah. And so another really interesting African mussel is over here. I'll pull this out so we can see it a little bit better. But this species is really interesting. It's a really fragile species. This one's from
that one is pretty cool.
Congo. And in some ways, it kind of looks like Utterbachia, something maybe you'd find in the
like our paper punch shell, kind of, but it doesn't have that cool shape there. It looks like from this outer space.
So it has this, this kind of flared. This is where like the siphons or the apertures are, where it's kind of flared out, and this is really unique to all freshwater mussels. And this is a representative of a family from the Iridienidae.
Only found in Africa, right?
Yes, that family. Yeah. Really fragile specimen, and it's amazing that its morphology, we've been able to take care of it.
Yeah.
And it made it here from Africa.
So we got the freshwater oysters, the Pterid, Iridienidae. What other family do we have, or do you have some on the table? Or
yeah, so we can look at the table over here, and there's a little bit closer to home.
So here in North America, there's 2 families, Unionidae and Margaritiferidae. We don't have any Margaritiferidae here in the Anacostia, but John will show some specimens of that family.
Yeah, so there is apparently a Margaritiferid in the Chesapeake drainage.
Oh, interesting.
I forget where exactly it is, but it's in the northern part of the
Pennsylvania.
Yeah, I can't remember what species it is, but yeah.
Cumberland? No.
I don't think it's that one.
No.
But I can't remember what it is. But that family is really unique. It's also very species poor, the Margaret Faraday. They're probably among the best pearl producing species for freshwater. And they're distributed only on the northern continents in like a very bizarrely distributed, like some in Alabama, and there's some along the mid-Atlantic, and then there's some in Vietnam and Europe. They're very patchy, very patchily distributed. But this is a representative of one. This is Cumberlandia monodonta. Its common name is the spectacle case. So you can kind of imagine putting a pair of glasses in here.
This one.
Yeah. And this is a really large specimen for this species. This one is endemic to Mississippi River, but mostly in the Midwest.
Yeah. And then there's some in New England, right? And then on the Pacific Northwest.
Yes, yeah, of this
okay.
family. Yeah.
And those use mostly salmon, right, as the host fish.
They use mostly salmonids, this family,
yeah,
migratory. This species doesn't use a salmon, which everyone thought a salmonid. It uses like oh, I can't remember the common name. It's Stenosioglossoform.
Yeah, got the common name of that.
Oh, moon eye.
Oh yeah,
moon eye.
yeah.
It's a type of fish that this species specializes in.
That's when you deal with scientists; they only know about the scientific name.
I will admit that I don't know. I forgot a lot of the common.
Yeah. And then here we got some other. Well, these are the rest of these are pretty much in the Unionidae family, so same family as the Anacostia River mussels. So this one is pretty cool. It's called the washboard. It's perhaps the largest species in the U.S. This one was collected in Eagle Creek in Kentucky. Look at the size of this thing. The size of my face. Yeah. And look at the juvenile. So these are the same, but they're younger specimen. Look at the cool morphology.
So yeah, you can imagine looking at the smaller ones, why they called this one the washboard. You know, it's much more corrugated. Whereas as they get older, they tend to lose some of that. This one has some interesting writing on the back. It was collected in 1919, and there's some measurements and weights. And then they have a note here that said, the weight of the shell, animal, and water must have been about 4 pounds and 8 ounces. So you can imagine having the other valve and animal inside of here pushing 4.5 pounds.
And this one, let me point out something else, especially if you have been in our volunteer events measuring mussels. We talk about the basic morphology. So these are the teeth. These are the hinge teeth, this part here, and this is other type of teeth. It's called the pseudocardinal teeth. And right here, these circular patches are what John was talking about, the adductor muscles that help the mussel open and close. Yeah.
So that's the washboard.
And then this one is pretty cool. Yeah. Oh, go ahead.
Yeah, this is the common name for this species is called the rabbit's foot, which I think it does resemble quite a lot. And it can be a really beautiful species. This is just a nice series of specimens showing what they look like as a juvenile and then up here to an adult. And all of these animals came from this lot, which means they were collected essentially side by side at the same place and time. And there's just a tremendous amount of variation, you know, from being very dark brown to light to green. And they have these really, this genus is unique, and it has this very beautiful pattern. They call it chevrons, those kind of triangles. This is a really special species.
This one is also listed, federally listed, Quadrula cylindrica, rabbit's foot. And then another one that we wanted to show is the Lampsilis hinsigi. So it's a type of lamb mussel related to our eastern lamb mussel, but this one is the Higgins pearly shell, I think it is, mussel. So it's a Midwestern species that was one of the first mussel species, or the first, I believe, to be listed in the Endangered Species Act in 1976. So just 3 years after the Endangered Species Act came out.
You can, maybe some of you recognize this too, but the animals on top here are the female. You can tell by this more square shell outline, and the one on the bottom is a male. This is the male. Yeah, and so do you want to talk about this specimen?
Yeah, sure. Yeah. So this is a yellow lamb mussel. So these specimens were collected where the Washington Memorial is, before the Washington Memorial was built in the late 19th century. So again, yellow lamb mussel, Lampsilis cariosa. We have not seen it in the Anacostia just yet, but it is common in the Potomac River. So same drainage.
Yeah, so these specimens were collected by a Smithsonian scientist in 1890. So you can see on the back of this label, I don't know if you can see this, probably not, but on the back of this label, it says collected at the dredgings on the Potomac Flats in 1890. And so I read this and I had no idea what the Potomac Flats were. So I looked into it a bit, and it was like the Potomac Flats is the area was this really big wetland in DC, and it was the area where like the Lincoln Memorial stands today, and the reflecting pool, and a bunch of the the monuments on the National Mall was this giant wetland. And so in order to reclaim that land, they were dredging the Potomac River and putting all that mud on the wetlands. And as they were dredging the Potomac, they were picking up all these live mussels and dumping them on Potomac Flats, the wetland. And so some scientists went over there and collected these animals. And so they kind of like helped tell the story of the evolution of the National Mall, like where those our most famous monument stand used to be just a giant wetland, and they filled that area in using the mud and clams from the Potomac River. So I really like how these specimens can kind of help tell that story, which, these specimens are critical for biodiversity and conservation and evolution, but a lot of them also tell a cultural story.
Yeah, so and basically, just to remind folks, this is the species that has the cool mantle lure to attract the host fish. Check on our website, and also the Chesapeake Bay program on social media. Share footage of our eastern lamb mussels doing the display from some of our mussels at Bladensburg Waterfront Park. So do check that out, which is one of the coolest thing in the mussel world and in the natural world, I would say. First time I saw that at Buzzard Point, I was by myself and I just yelled with emotion. It was one of the coolest things I ever seen.
And it is why I study mussels, and one of the reasons I think many people do. Yeah. So these specimens are some of the whole animal, and these are actually specimens that Jorge and I collected in our recent expedition to Costa Rica. So this is a representative of a family called Mycetopotidae, and so a different family than than the Unionids and the Anacostia. And this is also a representative of that family, and you can see why.
That's one of my favorites.
It has this really cool, so this is the foot. If you've ever opened up a Unionid, it looks very different than this, but the root mycete, like that's mushroom, and then poda is foot. So this looks like a mushroom foot, which how the family got its name. And
that helps them like sort of anchor into the mud, right?
Yeah, and so when we were like finding these in these really soft mud. You can imagine them sitting in the mud like something like this, and this foot's extracted down here. And when we were like grubbing around for them, they would pull their foot up and sink down further into the substrate. So those are two really cool representatives that occur in North America, but aren't the kind of the typical North American families. And this is a representative of Nephronius. Nephronius. Well, we don't know what species this belongs to, but Jorge and some colleagues of mine are sequencing the DNA of all these animals to help better understand their diversity and how they disperse south into Central America, or in these cases, how they disperse north from South America.
One little story about that I want to tell. So when we were in Costa Rica processing these samples, John found a mite in one of the mussels, and that's a mussel mite. And I've never heard about that until that day. So apparently there's mites that parasitize the mussel, live inside the mussel tissues. And that was mind blowing too.
Yeah, they're just like layering parasites on top of parasites. Yeah. So they're a really poorly understood group that has very few taxonomic experts. So if you're looking for a job, that could be a good one.
We need more mussel people.
And so maybe is there anything else you want to show over here? You want to look at?
Let's take a look at the. Oh, maybe just this one's the little like shark mussel. That's just my nickname.
Yeah. So these are mostly most of these are from Southeast Asia, which has been the focus of my research over the past 10 years. But recently, with Jorge and colleagues, I've started to work on Central American stuff. But this just kind of shows an assemblage of some Southeast Asian species. Some of them have these wings, they're called. Some others are really elongate. This genus is unfortunately named scabies, but it's a really pretty specimen with those elevated green ridges. They're really cool. Species from the Mekong.
So for a long time, North America was considered like the top hotspot for mussels, but recent research has shown that Southeast Asia has a couple dozen more species. So it's now on top, and North America is in second place in terms of diversity of river mussel species.
Yeah, they are very similar in species level diversity, but for a long time, they thought North America was, and it's because these Southeast Asian mussels were much more poorly understood relative to the North American stuff. But since we've been working on them and describing all sorts of new species, they've started to, even out in terms of their total species diversity.
It's something cool that John was saying. So this one is very similar to the one he just showed from Costa Rica, Mycetopoda, for different families, but they evolved the same shape in different continents because they use similar habitats, correct?
Yeah. And so like if you can imagine like the bivalve tree of life, this shape, this really elongate shape has evolved here and here and here. So independently converging on the same shape, just like the freshwater oysters, they are independently evolving the same shape as, oysters of Chesapeake Bay. And that process of convergent evolution has driven mussel biologists crazy for 100 years, because the animals look so similar, but based on their DNA or other parts of their soft anatomy, we know that they are very, very distantly related. Yeah.
So it's pretty cool. Yeah, like the cementing of the oyster on rocks evolved 3 times, right, around the world.
Yeah,
in different branches
of just freshwater mussels.
Just freshwater mussels.
Yeah. Let's go take a peek at this little display over here.
And while we're walking, I'm wondering if anyone from our audience has questions. You're welcome to unmute, or you can put your questions in the chat. See me yet?
Yeah, so basically here we wanted to show some of the historic uses of mussels, and so in the 19th century or before, they were used to make shirt buttons, and actually that industry drove a lot of species to extinction and to decline. There were like big plants in along the Mississippi, right? In Iowa, there's a big one, and there were people like whose job was to look for mussels for the button factories.
Yeah, so like Jorge was saying, especially in the Midwest, there was like this gold rush. They call it like the pearl button rush, and it like blew up this industry of pearl button. So they would have individuals grubbing in the rivers looking for mussels, and they would use these kind of drills to punch out these blank buttons, they call them, and then you can polish these. This is kind of showing the various stages. So like a rough cut blank button, and then finishing it, polishing it, cutting the holes. So they were a really important part of like early American industry, especially in the Midwest. And with the invention of plastics around like the 1920s, and with the continued decline of all these mussel populations, the pearl button industry started to collapse. Especially with the invention of plastics, it just became easier to build buttons with plastics than it was to grab them out of the river, drill holes in them, polish them.
Right.
It was very labor intensive. So this is kind of like not a totally uniquely American thing because it happened elsewhere, but kind of an oftentimes underappreciated connection between biodiversity and American culture.
And people normally ask us like, what was the use in the pre-colonial times? And what we know is that Native Americans used the freshwater mussels for eating, although as far as we know, it wasn't a staple. There were tastier things to eat like venison and turkey and whatnot and fish. So, but they used the shells a lot for different purposes, ornaments and utensils. And the same in other parts of the world. Actually, in our Costa Rica trip, we saw that some people eat them. It was in a very specific area near the Nicaraguan border. In the rest of the areas we visited, people had no idea the mussels exist. But yeah, there's similar stories from South America. In some areas, they do eat them, including like one of the oysters in Colombia is in danger because of that. And yeah. Oh, so, this is cool. You want to talk about the art there?
So we do have a quick question. And Mary is asking, what is the difference between a mussel and a clam?
So they are both common names, and they people use them very differently. So like if you were at a restaurant and you ordered mussels, those would be marine mussels in the genus Mytilus, and they're like smaller, and they're attached. They attach with these threads to like a hard substrate. Whereas clams don't attach, and these would, if you order clams at a restaurant or something like that, they are much larger, and they don't attach to it like a hard structure, and they're really distantly related animals. And then there's freshwater mussels, which is not a true mussel like the marine mussels. So and then in other parts of the world, like in Costa Rica, they wouldn't call freshwater mussels. They tended to call them
mejas.
Yeah, clams. Yeah. So they are used in a lot of different ways, which contributes to the confusion between the two names.
Yeah, I guess the only thing they share is that they are in the bivalve class, right?
Yeah, they're both bivalves, and they're on different ends of the bivalve tree. Yeah,
Yeah.
So this specimen is really interesting. This is a species Cristaria. It's from China, and so what they're doing is when the animal is living, they'll kind of pry open the two shells of the animal, and they'll surgically implant or just place these little plastic Buddha-looking pendants in a living mussel. And so then as the mussel is continuing to grow, it's secreting the mother of pearl, the shiny nacre on the inside of the shell. And so it's creating these little pearl Buddhas or pearl coated Buddhas. And they will sell these in souvenir shops. So a couple of these ended up in our collection. These ones are quite old. This is from 1897, so they were doing this 100 years ago, well over 100 years ago. And so just this kind of display here is just kind of showing some of the connections between humans and mussels.
Yeah, and because of these impacts and others, that's why mussels are some of the most imperiled group of animals, with 70% of the species being like extinct, rare, or threatened, or endangered. Whereas if you compare with mammals or birds, it's only like 12-15% of their species that are extinct, rare, threatened, or endangered. So it's a group that needs a lot of conservation help, and also it's a group that gives us a lot of benefits, not only to us, but the general ecosystems. Any questions? Have you, so folks there, have they volunteered with us in the past, or they're like new, they're sort of getting acquainted with our program?
We do have a question.
Uh-huh.
Do most rivers have freshwater mussels?
Yeah, freshwater mussels are really common on our planet. They occur in all sorts of almost any type of permanent liquid fresh water. So they occur in like really high northern lakes or really small tropical streams. So they're found on any continent except for Antarctica, although there are fossil mussels from Antarctica. So yeah, most streams and lakes, if they're healthy ecosystems, will frequently have freshwater mussels. Sometimes, like if it's really like acidic water, like associated with a bog or really black water, the water is too acidic to support mussels or most mollusk species. So those are clear exceptions. And then, of course, if there's not any fish in the lake, like some very northern lakes are fishless. And then if the host fish isn't there, the mussel can't be there as well. So, yeah, really a common component of most freshwater ecosystems.
Yeah, and all these mussel families depend on fish to complete their life cycle. There's some also differences in the larva. So some of like our mussels have a larva that have two valves, and they have sort of like some hooks to attach to the fish body. But in the other families, they might have only one valve, right? Lasidium. So they're called Lasidium instead of Glochidium. Our larvae are called Glochidium from the Unionids. Yep. Getting too much into the weeds here.
That larval morphology is really poorly known, but like unlike the North American mussels where they Glochidium, you know that form really well.
And we have another question. When we were shown shells from Africa, were the ones that looked like an oyster shell actual mussels?
Yeah, so those animals are part of the order Unionoida. So like when we say freshwater mussels, we're talking about the order Unionoida, which is comprised of 6 families. And all of those families are freshwater mussels. And so what makes a freshwater mussel is essentially just a couple of traits: that they live in freshwater, that they brood, that the females retain they have development within the females, and that they are parasitic. That's kind of the one trait that unites all freshwater mussels. The trait that makes a freshwater mussel. And those oysters are freshwater mussels. They belong to the same group that the animals in the Anacostia do, the same larger group. So despite having an oyster in their common name, they are mussels. Yeah. Again, the confusion with the common names. Yeah.
But it's pretty fascinating to know that there are freshwater oysters in the world.
Yeah.
Yeah. I definitely want to see a live one someday.
Me too. I've never seen one.
Yeah. And yeah, and like again, they all should have a host fish. I'm sure there's not quite as well known what kind of fish, but yeah, they in theory they use a host fish to complete their life cycle.
Yeah.
Another question. Parasite usually means detriment to the host. Do the larval mussels harm the fish host?
Yeah. So normally, because it's a parasitic relationship, it's not going to kill the fish. It does happen, but I think only in rare occasions, like in artificial settings, like a hatchery, if they put too many larvae in a small fish, then it can compromise, the gills and the respiratory system, and in that case, the fish dies. But I think in nature it might be rare, right?
Yeah, that's my understanding too. The infection rate is so low that it does cause stress to the animal, probably like mostly respiratory stress. But like most parasites, you don't want to kill your host, because that's the only way that you're reproducing or continuing with your life cycle. So they are true parasites. They do harm their hosts, but it's relatively minor in the wild.
Yeah, and the one difference, so in the Anacostia and Potomac rivers, you might have seen the Asian clams, which are small and kind of roundish. So those clams, different with our native mussels, is that they don't need a host fish. So they just throw the larva, and the larva goes into the water column, and wherever it lands, it starts growing if it's the proper habitat. That's why we see a lot of clams. Actually, the clams outnumber the mussels in our rivers here. Yeah.
Okay. Another question. Are the habitats you see with South Asian mussels similar to the habitats we see in DC?
Yeah, in a lot of respects they are. They occur in really small creeks, and then larger rivers like the Potomac, or even larger rivers like the Mekong, Mississippi. And yeah, they're living sometimes in rocky habitats, sometimes in muddy habitats. So yeah, they're kind of occupying the same similar habitats.
Yeah, and like in the case of the Anacostia and Potomac rivers, you might see that some species like the paper bone shell, they really like the mucky habitats, whereas other species are going to be more numerous in the more gravelly and less mucky, like the northern lance, which is the very lance-shaped mussel we have in the river. And yeah, it depends on the species. So again, we have 8 native mussels in the Anacostia River, around 16 in the greater area, and in the US, 300 and something species. Yeah. And southeast and the southern area of the country having the highest diversity. Alabama, the state of Alabama has over 180 species, just the state of Alabama, which is pretty remarkable. Yeah.
Okay. One more question. So do marine mussels use host fish?
They do not.
No.
Yeah. This parasitic relationship where the larvae are parasites of vertebrates is unique to this group. So like the mussels that you get at a restaurant are free living animals. They are non-parasitic.
Oh, and one cool thing that John mentioned, like in the marine ones, they have the Bivalve threads. These are protein. They look like hair that helps them attach to surfaces, and that's how they grow them. They put them in these bowls, and they grow on top of the surfaces. Now, our mussels don't have that, but they have the vestigial Bivalve threads. At least our land mussels, I remember right now, when they are really tiny, when at the hatchery, at the Harrison Lake hatchery, our partners there. So when you grab a bunch from the sand of the tiny ones, I'm talking about like 1/4 of an inch little baby Eastern line mussels, they get attached to your fingers in little like threads. So those are the vestigial Bivalve threads, but they lose that capability as they grow.
And I'll make another call for any questions anyone has in the audience. We don't currently have any.
This is your opportunity to learn about bivalves and mussels. And again, check out our many, we have a lot of volunteer opportunities for mussel monitoring of mussels that come from the hatchery. Mussel surveys to check our wild mussels in different spots from Buzzard Point, Kingman Lake, Canover Lake, and mussel release event tagging and or release events, which are pretty fun for the public, and check out also through the schools where we work with the Mussel Power education program. And check out, do we still have that shirt, Keisha, on the pop-up store?
Yes, we do,
Yeah.
in our bonfire shop.
Bonfire shop, we have one of the designs by a high school student, which is pretty cool, the Mussel Power.
Yes, it's very cool. Yes, it's available in our bonfire shop. If you go to our Facebook page, the link is on a recent post. And also if you go to our website, you'll be able to find it. And we do have a few more questions. So first question, is your collection mostly mussels we no longer see, or more so of mussels that are still being found in nature?
Yeah, so we do have representatives of extinct species. There are relatively, we do have extinct animals, which is unusual for an invertebrate. Most marine vertebrates are not as extinct or susceptible to extinction as freshwater mussels. But the vast majority of our specimens are from extinct or living species. But we have a really nice collection of extinct mussels that were collected in the early 1900s, but then went extinct, in the 1950s or something like that. So we have a really strong collection of extinct species, but most of them are extant or living species.
Okay. And the next question is, how do mussels filter toxins?
Yeah, why don't you give it a shot?
Yeah, so basically as filter feeders, they pretty much filter whatever's in the water cone. And from there, they assimilate what they find nutritious, which could be a plankton, like a microscopic algae, protozoan, even fungal spore or bacteria like E. coli. And that's what they eat. And then what they don't eat, they expel it, which is, let's say, sediment and stuff like that. So that's why they help the sediment settle. Now, if the pollutant is like, if it's like a heavy metal or too much ammonia, they will just die because they don't have the capability of processing those things. So sort of it depends on in other words, if the chemical is too nasty, they will die. If it's something minor, like they might still process it.
Okay. And being dependent on a host fish just seems so limiting to general survival. What's the evolutionary advantage?
Oh, it's a really insightful question. And so being a parent, this is a really complex life history, which seems to be limiting, but this group of animals is like the most successful radiation of freshwater bivalves. So bivalves have colonized freshwater multiple different times, but it's because of the parasitic relationship that has allowed them to diversify. The advantage of the parasitic relationship is, as you might imagine, these animals don't move around very much as adults. But this parasitic relationship allows them to use their hosts as vectors for dispersal. So like freshwater mussels are distributed across the planet in the innermost parts of the continents and in almost any type of habitat. And it's because of the parasitic relationship that allows them to have greater dispersal potential. They're just using the mobility of a host, their fish, and they act as hitchhikers to, hijack the mobility of fishes. So what seems like a complex life history that makes you susceptible to extinction, that is true, but it is the adaptation that allowed them to colonize the continents and diversify in rivers around the world.
Yeah. And also, I would like to add that, for example, here in the Anacostia River, the Potomac River, we've got migratory fish that serve as host fish for these mussels, and these fish move a lot. They move hundreds of miles. So if you think about it, these fish species are a great way to move around. Like you think like alewife, shad, which are the host species of our alewife floater, or American eel is the host fish of Eastern Olympio. So great transportation for the mussel larva.
Exactly. Yeah.
Whereas others, like mummy chog or introduced carp, they're gonna stay in the same area. So perhaps not as great, but the migratory fish, yep.
Okay. The next question: do you presume that the mussels that went extinct in DC was because of the dredging?
So I don't know if there has been any confirmed extinction in the Potomac. There are many species that haven't been found or confirmed in decades in the Potomac. So I wouldn't call them extinctions. You could maybe call them population level extinctions.
Yeah.
That's also called like extirpation. But while the dredging definitely, removes a lot of animals from this population and destroys habitat, it's kind of probably a minor, I don't want to say minor, but like, long-term pollution or
Damming,
the restriction of the host fish are probably more consequential than the dredging. While it has huge impact, especially in the short term, in the long term, like terms of like population stability, probably less important.
Yeah, and actually that was a shock to us because people asked us I've been at AWS for 15 years and people before 10 years ago asked me mussels in the river, and before I didn't know anything. I learned on the job pretty much. I used to sort of dismiss mussels in the Anacostia, but then we started finding them as we did like submerged aquatic vegetation, wetland work, and because the Anacostia River is tidal, it's a very stable environment. So even with all the neglect and pollution in the last couple centuries, we still have 8 species of mussels, and places like Buzzard Point and the lower Anacostia have very healthy mussel beds, surprisingly. And it is actually when we go in the upper part of the watershed in the non-tidal streams, we haven't done an assessment, but as far as we know, there's not many mussels. Why is that? Because it's not very stable. When it rains, say half an inch to an inch, water gushes, and so it's not a very stable environment. That's why there's not many mussels there as far as we know. Although recently we found shells of Eastern Olympio and Eastern Floater in the Upper Beaver Dam Creek, which is a huge property by the USDA. So it has been protected there for almost 100 years. So forests and agricultural land. So it's as pristine as you can get in the Anacostia watershed. And yet we found shells and mussels. We want to go back with the Maryland DNR folks and assess the area, but it could be like the only population outside of the tidal environment.
Okay. And I feel that our next set of questions have been answered, but see, Kel, if you are still and you have any other questions, please go ahead and put those in the chat, or anyone else.
In the meantime, I would like to mention that one of our long-term goals, and we're talking to partners, is to map all those high-quality mussel beds in the lower Anacostia and the Potomac in BC, because we observe them, and a lot of people are amazed, including I brought Jess Jones, a specialist from Virginia Tech, and he was surprised to find that many mussels here in a city. He's more used to the Tennessee River and more pristine areas of the country. So the fact that we have these healthy mussel beds is pretty amazing, and we should do as much as we can to protect them, because they are doing all those ecosystem services, biofiltration, and a lot of stuff. So it's like when you compare like a created wetland versus a natural wetland is always going to be the best in terms of the benefits. The created wetland is going to take years to get to that point. The same with our mussels. It's great that we're propagating mussels, and they'll get to a point where perhaps, hopefully, they will get like what we see at Buzzard Point. But right now, we should do as much as we can to protect those healthy mussel beds in BC.
And another question. Do you have concerns that dredging of the Anacostia River might be detrimental to mussels population because of, for example, increased river depth?
Well, there's going to be some impact when they do the remedial actions for sure. That unfortunately, it's going to have some impact in places like Kingman Lake, where we have actually released some mussels. Although recently we're mostly releasing at Kenilworth Lake, because that won't see any remedial actions. Some mussels are safe there, but at Kingman Lake, there's a couple of like narrow tidal streams about the width of this hole right here. So we think like a salvage is more doable instead of the big open river. But yeah, in areas of the main stem, the open river, unfortunately, there's going to be some impact. But I look at more like we're going to have a big opportunity once the remedial actions are done. That's going to be a lot of habitat for mussel propagation in the future. So I always tell people this is just the beginning. What we have been doing since 2018, we have released more than 36,000 mussels of 4 species, but that's just the beginning. I mean, after the remediation, we're gonna see a lot of opportunities. And as for the changes in depth, I don't think it's gonna be a significant changes in depth to damage in other words, it's not going to be as deep to be to make it a worse habitat for the mussels.
And next question: is there any data on how far a mussel will move in a year?
There's been some research on like individual movement. I don't know. I wouldn't venture to guess like a number, but I know there's been some research. The first one that comes to mind is kind of like the vertical migration, how individual mussels are moving up and down in the substrate between like summer and winter in like northern lakes. And so they don't move around all too much up and downstream unless they get blown out, like by if there's a really large flood or something like that. Sometimes individuals will get washed away and oftentimes not die. So there has been some research about mussel movement, but to be perfectly honest, I don't know all too much about it.
Yeah, I know that in some streams in Virginia, they find them like mile downstream, but it's because they get washed. In an environment like the Anacostia, the tidal Anacostia, they don't move that much. And we see like the pond mussels in the spring when they are brooding, the females are out and about. They move a lot, but it's just, a few feet. Now, in depth wise, I had an interesting experience. One time we were collecting American lotus rootstock at the Mattawoman Creek in the Potomac. And I had my whole arm. It was early spring, so late winter, early spring. So I was looking for roots and I had my whole arm in the mud. And I found an Eastern floater, but it was sort of in wintering mode. But that as deep as I have found them. Some of the mussels that we released at Kingman, we put the radio tags, and then you come with a thing that looks like a metal detector and find them again. We were detecting some animals, but when we were raking with our usual the sampling rakes, we couldn't find them. So they were, and that was in midsummer. So yeah, who knows how deep they were at that time, but yeah, they do bury in the mud.
Okay, a connected question. What is the pattern of vertical movement seasonally?
Yeah, I think in the winter they're gonna go deeper for sure. Or if conditions may seem like unfavorable during the summer, like flooding or the water has too much nasty stuff, they might just hide or shut, bury and shut. Yeah. And actually that's what they do when water conditions are not good, they shut. That's why in some places like Minnesota and recently I heard in Poland too, in a water treatment plant, they put some sensors in the valves. So when they shut, that means something's wrong with the water. So I thought that was pretty cool with that approach.
Like an early detection method.
Yeah.
Bivalves inspired early detection.
Yeah.
Things to recover.
Are you seeing distribution of specific species up and down the river?
Yes, so we've been doing surveys since 2017, and we have found some interesting things. Now, these are qualitative surveys, and but we have found, for example, at Kenilworth Lake, we see more and more mussels, and that's one of the places where we have released mussels. And also another place is Kingman Lake, right by Heritage Island. It's a place where we didn't find a lot of animals and a few species. But in the last years, we've been finding a little bit more and more. One of my theories is because that place has seen an explosion of submerged aquatic vegetation. So the submerged aquatic, the underwater grasses are great for fish. So if there are fish, they are chances are you're going to find mussels. So in places like Buzzard Point, we found that that's like the hotspot. So just to give you an example, in 1 hour, we can find 220 plus mussels at Buzzard Point. At Heritage Marsh or Kingman Lake, 10 to 20. The most we have found in the upper Anacostia is at Kenilworth, where we've been seeing more mussels. The last year we found over 80 mussels in all our year surveys. So we've been learning like where they are, where these different species are, and what type of habitat, and that helps us also with the propagation. Like this year, I didn't have to snorkel in the spring in cold water. We looked for the pond mussels by wading, just with waders, because we know that at Kenilworth we have a lot of them. So we didn't have to snorkel at Buzzard Point like I've been doing for the last several years in 40-degree water. So that was great. And actually, like our partners from the Maryland DNR, they think that this could be one of the biggest populations of eastern pond mussel in the Chesapeake Bay region. So that's pretty cool that we found that with our community scientists in our surveys.
Okay. Because you have specimens, are you seeing any differences in the shell specimens that we see today compared with the same species from decades ago, for example?
Some folks have done some really interesting research on that using historical specimens and comparing them to modern ones. And one of the things that they have found, this is from research in the Midwest, that for mussels of similar age, you can age a mussel much like you can like a tree ring, using tree rings. So for mussels that were the same age, the historical mussels were smaller than the more modern day, the ones in the river today. And so they're growing faster, essentially. Using historical data, people have determined that modern mussels are growing at a faster rate than they used to. And this was done in the Midwest, where there's lots of mono agriculture. And so there's lots of mussels, some mussel species are doing very well there, but there's lots of additional nutrients in the river. So they think that that is causing mussels to grow larger faster. So that's one of the ways in which historical collections have been used.
Which brings me to another topic, like in our monitoring of hatchery raised mussels in all these years, we found that mussels in the Anacostia River of the same species grow more than mussels in the Potomac, deployed in the Potomac River. And we think that is because of the nutrients. And now the Anacostia is a smaller system, so it's more concentrated. You know, you get input from a lot of the tributaries like Northwest Branch, Northeast Branch, and it's also especially the middle section is really, really sluggish. So the water doesn't move a whole lot. It has to be like a huge storm to make it really flow down river. So that's why it's sort of more concentrated, more nutrient rich than the Potomac. That is just a bigger system. And when I said Potomac, we mostly around Blue Plains Water Treatment Plant, JBAP, the Air Force Base. That's where we had mussels in the Potomac.
All right, and the last question we have, do mussels occur more in moving water than in ponds or lakes?
Both, really. They occur in both ecosystems. And one interesting thing, and also talking about nutrition and such, perhaps, for example, eastern floater is one of the most common species. The biggest specimens I've seen in the watershed are at Greenville Lake. One time they were dewatering the lake and we had to salvage some mussels, and those eastern floaters were giant compared to the ones we find, let's say, at Buzzard Point, which sometimes we find a big one, but not as big as the ones that we saw at Greenville Lake. And we think because that lake is fed by a creek and storm water, so it's really nutrient rich. So, you know, these mussels are eating like McDonald's every day. So yeah, that's one of the differences I've noticed.
Okay. And we don't have any other questions.
All right, so thank you so much for joining us today. It's a very special opportunity here with our partner, John, at the Smithsonian Institution. And yeah, check our website, Anacostia WS, and social media at Anacostia WS for more opportunity. And John, thank you so much for this. It's always nice.
It's my pleasure. Thanks for joining. Maybe I'll see you out on the river sometime.
Yep.
I'd like to join you guys.
Okay, great. Thanks everyone for joining. I'm going to go ahead and end.